We start with the bacterial cell. Okay, the chapter that we are doing is cell structure. Okay, so we are going to first start with the bacterial cell and then we'll come to the other cells. See, the other points with regards to, the names of the scientist and all, that is all to be read by you. Okay, so I'm directly coming to the concepts. So when it comes to a bacterial cell, okay, so but before we do a bacterial cell, what is very important is you should know this first division of what is a prokaryotic cell. Okay, what is a prokaryote and what is a eukaryote. I assume that you all know what is prokaryotes and eukaryota, right? Now, pro is nothing but, and carry on. Carry on means a nucleus, okay? A carry on or karyota is a nucleus. Pro means they do not have a true nuclear membrane. So they do not have a true nuclear membrane. But when it comes to eukaryotes, the meaning of eu is true. Okay, they have a true nuclear membrane. So coming to a cell, I hope you all know a typical cell, okay? So what you have is, okay, a nucleus at the center, okay? And you have a nuclear membrane. So this is your cell membrane outside. So when it comes to prokaryotes do not have well-defined, nuclear membranes as such. They do not have a well-defined nuclear membrane, which means if I speak of a bacteria, example of a prokaryote is a bacteria, okay, all these bacterial cells basically. Now, when it comes to bacteria, the structures what we are discussing now, they do not have a nucleus. So basically the entire DNA, I hope you know that in a eukaryotes, there is a nucleus right at the center, okay, and all your DNA basically they are arranged in the form of chromosomes, right? So there are chromosomes which are present here. Your entire a nuclear membrane is also present around, but there is no nuclear membrane in a bacteria. So entire DNA is just lying coiled in the cytoplasm. Can you see that? So your entire DNA is just lying coiled in the cytoplasm. So they do not have any other organelles which have a well-defined membrane as such. Now I'm sure you know for mitochondria, chloroplast, right? All these cells basically have a well-defined membranes around them. So they do not have their organelles, okay? The organelles which are present in the bacteria do not have well-defined membranes, whereas eukaryotes do have it. So I expect you to read, a little more points with regards to which organelles are present in prokaryotes and which are not present. Okay. So this division, this differences is what you will have to know. We're discussing a prokaryote and eukaryote. Based on that, you can, then start reading about what are the differences between them. So let's start with prokaryotes. All right. Now, coming to prokaryotes, basically here, like, when it comes to eukaryotes, they do have, okay, all the organelles here do have, here they do not have. Here the organelles have well-defined membranes. All right. So let's come to the structure of a bacteria first. So your first learning is a bacterial cell. Okay.
Ma'am, please scroll.
Yeah.
Thank you, ma'am.
Yeah. Okay. So let's directly start with a bacterial cell wall. Okay. Let's start with the bacteria. Now, when it comes to a bacteria, first, okay, let me draw out a rough picture here. So if you can see a picture of a bacteria, right? This is what I'm drawing out for you. Cell membranes, I'll come to it a little later. Okay. So what you have is a flagella, which is a tail. Can you see this flagella? This is a tail. This is actually needed for movement. For propelling. This is a propelling organ. Okay. Which is movement, to move ahead or to move like, in a particular direction. Now, as I said, the cell membranes are here. Okay. The cell membrane. There is no nucleus as such. So the entire DNA, can you see it's a double-stranded DNA? It is a double-stranded DNA, which is just lying in the cytoplasm. Now, this cytoplasm, what you can, this DNA material is called as the nucleoid. It's called as the nucleoid, also called as the gonopore. also called as the gonopore, which is just lying in the cytoplasm. All right. Now, what these, bacteria does not have mitochondria and chloroplast as such. They do not have it. But if you look at the inner membrane, okay, I'm drawing the inner membrane of the bacteria in red. This inner membrane is folded to give you some rough folds as such. So this entire inner membrane of the bacteria is folded to give you, a highly folded region as such. This is called as the mesosome. This is called as a mesosome. And as I said, a bacteria does not have of mitochondria. So the role of mesosome is that it helps in respiration. Okay, so all the respiration is nothing but utilization of glucose. Okay, so glucose in the presence of oxygen will give you ATP, correct? That is respiration. Glucose is broken down in the presence of oxygen to give you energy. But there is no mitochondria here. And I hope you all know that mitochondria helps in formation of ATP. So since there is no mitochondria, what they have is mesosomes. And mesosomes act like a mitochondria. Okay, they help in ATP formation. So that is what I said. They help in respiration. They help in respiration. Okay. Now, next, what you should know is, if you look at a bacteria, they do have ribosomes in them. Ribosomes are organelles. I hope you know that. Ribosomes. But ribosomes in a bacteria, okay, they are basically present as polyribosomes. We call them as polyribosomes because they form chains as such. They form chains as such. So these ribosomes are present. Okay, they're usually present as ribosomes, polyribosomes. Have a look at this picture. Ribosomes here, okay, but they're usually present as polyribosomes, okay. The cytoplasm is here, right at the center, okay. Cytoplasm is this fluid in which all these, ribosomes and structures are present. Now, next structure that you should know of is, certain bacteria have a structure called as pili. Okay, they have structures called as pili. Now, pili is made up of a protein called as pilin. Pilin is the protein, and flagella is made up of a protein called as flagellin. Flagella is made up of a protein called as flagellin. Pili is made up of a protein called as pilin. Now, what is the role of pili? What is the role of this pili? Now, pili basically, when it comes to the structure, when I speak of a bacteria, okay, so here I'm giving an example of two bacteria. Okay, so bacteria, basically they're not like humans. They're very friendly to each other. Okay, so what they can do is, using their pili, if you see here, pili of both the bacteria, they fuse together. Okay, they fuse together and they end up giving you a pilin tube. Look at this. So pili of both the bacteria, they fuse together, and what you end up getting is a pilin. This is a pilin tube. Okay. Now, what can happen is, if this bacteria here, okay, has got a DNA, double-stranded DNA, can you see here? If this bacteria has a double-stranded DNA, I call it as the F+ strain. And this bacteria here does not have, okay, let's say it does not have some genetic material in them. Now, what this bacteria can do is, this bacteria can give one of its DNA strand. Can you see here? It can give one of its DNA strand, which can be released, via this pilin tube, and it can enter the F negative strain of the bacteria. Okay, so it is giving off one of its strand to the neighboring bacteria. How? Via the pilin tube. Okay, this pilin tube is also called as the conjugation tube. So make a note, this bacteria has given one of its DNA strand to the neighboring cell that it is fused with. Now, using one of its strands, this bacteria can make one more DNA strand over it, and this one also can make one more DNA strand over it. Okay. So again, both of them. So basically, what is the meaning of, what is the use of this pilin tube? One bacteria can share its genetic material with the other bacterial cell via the pilin tube. So this process, okay, now what is formed here is This process is conjugation. So what is exactly conjugation? Conjugation is a process. It is not sexual reproduction. Bacteria do not reproduce sexually. Okay, so it is not sexual reproduction. It is a mistake if it is, if it is given in your book. It is not sexual reproduction. Bacteria do not reproduce sexually. They reproduce only asexually. So what is conjugation? A process by which bacteria transfer the genetic material via a phylum tube. Okay, so this is not mating. This is transfer, giving some genetic material from one cell to the other cell. Have you understood this?
Ma'am, what is the reason for not undergoing sexual reproduction?
Bacteria, they do not have the genetic material. So basically, they do not make gametes. Okay, so the progeny, for them, it's better, to have their progeny which is similar to them only. Why is because by these processes they can have variations amongst themselves. Okay, now see, in humans there is sexual reproduction because of which there is variation. We are little highly evolved organisms. They don't have the genetic material. They're simpler forms. They don't have the genetic material in them, okay, to undergo differentiation as such. Because of which they adopt to a very simpler mode of reproduction, which is sexual mode of reproduction. They don't have the genetic material. They don't form gametes, no. Hence, no sexual reproduction. Got it? They're simpler. They evolved like that. They have not yet, evolved to become higher organisms. They don't form gametes. They don't have the genetic material in them. First of all, such a small little structure, DNA present in the cytoplasm. But they can bring about variations like this. They can transfer, genetic material from them to the other cell. That is how they can bring about variations. That's why you can have different species of bacteria. Okay, so one bacteria can give off its genetic material to the other and bring about changes in the other bacterial cell. You get it?
Yes.
Okay. Has everybody understood this part? Phyli.
Yeah.
Okay, now let's come to one more structure which is present on the cell membranes. Okay. Now these are called as fimbriae. Fimbriae is different, okay, than phylum. Fimbriae. Now, fimbriae is needed for adhesion. Adhesion means attachment. They need for attachment. All right. Now, bacteria, when they get into your body, they have to get attached, to some surface. For that, fimbriae is useful. Okay. Have you understood the difference between phyla and fimbriae? Phyla is not for attachment. This is also called as a sex phyla, but it is wrong to call them as sex phyla because they are not involved in sexual reproduction. It's just to transfer the genetic material. Okay, let's come to the next part. The cell membranes are very important. Okay, so what I'm discussing now is some more structures. The cell envelope I'll come to. Now, second thing you should know, what is a cell envelope? Okay, so cell envelope is nothing but glycocalyx plus cell wall plus plasma membrane. This together is the cell envelope. Okay. This together is the cell envelope. Okay. So this together forms the cell envelope. Now this basically cell envelope is protective in nature. Protection. Okay. So let's come to glycocalyx first. Now, glycocalyx could be, this is the outermost protective layer. Okay, it's a viscous layer which is present outside the cell wall. So I said that this is the cell wall, right? Okay, or cell membrane basically. So I'll tell you like these structures first. First outer layer, what you're going to have is a glycocalyx. This is G for glycocalyx. Okay. Then you will have cell wall. In bacteria, we use the word cell wall, which is just like the cell membrane only. And then I'm going to have one more layer here, okay, which is going to be my plasma membrane. Let's come to glycocalyx. Now this glycocalyx could be of two types, okay. The glycocalyx could be of two types, A and B. Now A could be, it could be two types. One could be a slime layer. The glycocalyx could be slime layer, or it could be a capsule. Okay. It could be a slime layer or a capsule. Now, when it comes to slime layers are viscous in nature. Okay, they are viscous in nature. And it consists of polysaccharides. They are all polysaccharides. Glyco means, you should know, glyco means polysaccharides. Okay. Now, this entire glycocalyx, be it slime layer or the capsule, okay, they help in protecting the bacteria from antibiotics and desiccation. So the entire glycocalyx, if you see, okay, glycocalyx, it is protective in nature, correct? So they help bacteria against antibiotics. Against antibiotics and desiccation. Desiccation is basically like, for example, when a bacteria is exposed to extreme conditions, there are all chances that the bacterial cell wall could be degraded. Desiccation means, preventing water loss from the body. Once the water loss, there is water loss in the bacteria, it's dead. Okay. You can consider it to be dead. So that is desiccation. They help or they prevent the bacteria, okay, from antibiotics, action of antibiotics and desiccation. So if a glycocalyx is present in any bacteria, no, it increases their power of infection. So they happen to increase the power of infection. So any bacteria which has glycocalyx, it's difficult to basically get rid of them or kill them. Example is this Bacillus anthracis. Okay, Bacillus anthracis, which causes anthrax. All right, let's come to cell wall. Understand the cell wall well, okay? Let's come to the cell wall. Now, cell wall of bacteria is made up of peptidoglycan. Don't ever forget this, peptidoglycan, which is also called as murine. Okay. Also, they are type of mucopeptides. This we have done in polysaccharides, right? There are also mucopeptides. Obviously, this is present, I told you, below the glycocalyx. Cell wall is present below the capsule. okay, or the slime layer, but outside the plasma membrane. This is also needed for, maintaining the shape and internal structures. This is also needed for maintaining the shape and the structures. Now, based on the cell wall, okay, based on the cell wall, a bacteria can be gram positive or gram negative. I'm explaining this part to you. A bacteria can be gram positive or gram negative. Now, Sir Christian Graham, he was the one, okay, who came up with the gram staining technique. He came up with the gram staining technique, okay. And based on him, we use this, based on him, like, his technique, we can first differentiate bacteria into gram positive or gram negative. All right. Is it understood till here, students?
Yes, ma'am.
Yes, ma'am.
Okay. Now let's come to this structure here, gram positive or gram negative. Okay. Now you see, basically, this gram stain is made up of crystal violet. This gram stain is nothing but I'm using a stain which is called as crystal violet. Okay. So what I do is first I take a bacteria, okay, on a petri dish or a slide. Let's say I'm taking bacteria on 2 slides. What I do is first step, I add crystal violet. Okay, I add crystal violet to both the bacteria. I dip them. So I'm taking it on a slide and I add crystal violet, CV for crystal violet. Then I wash them. Okay, I wash it. What I do is then I add iodine. This also I'm adding iodine. Next, what I do is I add safranin. And then I wash it. Okay, then I wash it. Let's say after doing all these steps, I see that this bacteria is, purple violet in color. This is purple violet in color. And let's say this is red, pinkish red in color. Anything which is pinkish red, which means it has picked up which stain? That means it has picked up safranin stain. And if it is crystal violet, that means it has picked up the violet stain. Are you getting it? So such a bacteria will be called as gram positive. That means the ones which appear violet in color will be gram positive. The ones which are pinkish in color will be gram negative. Okay, so a rough step I've told you as to what is to be done, okay, when it comes to staining. Now let's come to the principle which is involved here. Just let me know once you're finished. Now, okay, when it comes to a gram positive bacteria, now what they have, just see, they have a thick peptidoglycan layer. Can you see this? This is a thick peptidoglycan layer, this full from here to here. Okay, this full structure, peptidoglycan layer. And inside they have a plasma membrane. So peptidoglycan is a part of what? Cell wall. You get it? Now look at this gram negative bacteria. They have a very thin peptidoglycan layer. If you see just right inside, only here, only this small layer is peptidoglycan. Okay. This small layer here is peptidoglycan. Outside they have an outer membrane and inside they have an inner membrane, outer and inner membrane. And in between they have a peptidoglycan, which is the cell wall part. Okay, and these are plasma membranes, outer membrane here. Inside is a plasma membrane. This composition, this outer membrane, I'll tell you what this is. Okay, just hold on. So, reason why a gram positive bacteria, I told you, will appear purple, correct? Gram positive is purple, violet is because they retain what? They retain which stain? They retain crystal violet stain. Now, why do they retain crystal violet stain? Is because this thick peptidoglycan layers here, can you see? They actually happen to retain crystal violet stain. So crystal violet basically comes and binds to the peptidoglycan layer. Okay. And iodine will help in fixing of the crystal violet. So iodine helps in, it's a fixative. Okay. Iodine will help in fixing. But if you look at this gram-negative bacteria, peptidoglycan is layer is inside. So even if crystal violet comes here and binds, iodine cannot help in binding it to the outer layer because this has to penetrate in, correct? But obviously this layer outside will not allow crystal violet to penetrate in. And iodine also cannot help in fixing it because crystal violet is bound to the outer membrane. During a wash procedure, all of this crystal violet gets washed off. But here, even after a wash procedure, crystal violet will still remain is because they are tightly bound to the peptidoglycan layers. Okay. So next step, what we are doing is we are adding safranin. So when I add safranin will nicely come and bind to the outer membranes, and because of which this bacteria will appear pinkish, pink red, because it is going to pick up safranin. Here, this is not going to pick up the second stain because it is already stained with crystal violet. It is not going to pick up safranin. Hence, it will appear violet in color or purple. This will appear reddish pink. Have you understood this part? It's all because of this peptidoglycan layer. This is inside, right? Here, the peptidoglycan layer is inside, and a thin one. So I hope you have got the difference between gram positive and gram negative. Have you?